Hi, my name is Sylvere Bastien. I am a bioinformatician at CIRI located in Lyon, in the staphylococcal pathogenesis team led by Professor François Van den Esch. I'm going to talk about an ongoing project regarding the development of an automatic pipeline using nextflow for the selection of peptide sequences in targeted proteomics. In collaboration with the Institute of Analytical Sciences, we have implemented a high-throughput targeted proteomics method. The goal is to be able to quantify any allelic variants of proteins of interest using short protein sequences. Following the specification defined by the proteomic researchers, we needed to generate peptide sequences that must follow certain conditions as described here. Therefore, we have developed an automatic pipeline capable of constructing a list of variants of a targeted protein from a potentially large protein database. Our aim was to create a fast pipeline with memory consumption suitable for any computer while adhering to FAIR principles. This pipeline only requires two mandatory inputs: a folder containing a set of amino acid FASTA files and a protein sequence of interest. The first step is called database preparation. This stage involves generating a compact database while retaining the entire genomic variability. Some of the databases can be large and not curated, so it was necessary to limit memory consumption in order to make it usable on any computer and to control the sequences. The second step aims to select all allelic variants of any protein of interest. One of the major challenges is how to select the correct set of allelic variants compared to distinct but highly similar proteins. To address this problem, we decided to use a funnel-like approach with increasingly stringent filters. In some cases, we discovered that some allelic variants were extremely rare, so we added a user-defined parameter to keep only a subset of these variants, based on the frequency of each allelic variant compared to the total frequency of variants present in the database. The third step selects the final list of peptide sequences. This list should be as small as possible while ensuring that each variant is covered by a user-defined number of peptide sequences. These are generated by simulating the usage of trypsin hydrolysis on the selected sequences. Peptides that are found in multiple copies in the initial database are removed, and an option has been implemented for the user to add or delete specific peptide sequences. Finally, the last step, called peptide validation, checks the specificity of the final list of peptides. The goal is to list all the sequences which contain at least one peptide derived from the initial database. Here is an example of what can be obtained. A few words about the database. We retrieved approximately 66,000 SRS FASTA amino acid sequences from GenBank. This example represents results from ECCB, which is an important valence factor of SRUs. The final peptide list contains 3 peptides and was generated from 18 allelic variants, representing 98% of the total frequency of all allelic variants of ECCB. With these 3 peptides, we can quantify 18 allelic variants as well as 26 other variants that accounted for the remaining 2% in terms of frequency. We can observe that if we decrease the threshold of the number of peptide sequences to detect in mass spectrometry, we will quantify proteins that are no longer allelic variants of ECCB, but variants of other proteins. In conclusion, This pipeline has been validated for 41 virulence proteins in SRAs. It has also been used for the future detection of virulence proteins in E. coli and P. aeruginosa. It helps in selecting peptide sequences for the specific detection of any proteins in any species, including genomic diversity. The entire pipeline is available on GitLab, and the use of nextflow and Docker ensures reproducibility of the results. Regarding memory and time consumption, and as I said, the database contains 60,000 genomes, and we can see that the pipeline remains within a run-suitable for any computers. I would like to thank all the people who participated in this project, and especially Pauline, Sarah, and Julia. If you are interested and would like more information and details, you have the QR code to access the poster and the information to come and see the poster in Lyon. Thank you, and I wish you a great conference.